Well, hi everyone. Welcome to Quantitative Seminar. I'm Zoe, and I'm one of the Quantitative Seminar organizers here. So I'm going to start off with a land acknowledgement. The University of Washington acknowledges the Coast Salish peoples of this land, the land which touches the shared waters of all bands and tribes in the Suquamish, Tulalip, and Muckleshoot nations. Our lab also acknowledges that we live and work on the ancestral lands of the Suquamish people, the first people of Seattle. And I'm really excited to introduce our speaker today. Ingrid Spies has worked at the Alaska Fisheries Science Center for 20 years in a variety of positions, from genetics lab tech to stock assessment scientist. She received her master's from SAFS in 2002 with Paul Benson and Karen Naish looking at the origin of Lake Washington sockeye salmon using genetics. She completed her PhD in 2014 with Andre Hunt in a project that combined the fields of population genetics and fisheries management. Her current work combines her genetics and stock assessment background to tackle complex problems in fisheries science that often involve issues with spatial management of commercially exploited Alaska groundfish. She has published on identification of several new species and genetic issues, including stock structure and phylogenetics in Pacific cod, Atka mackerel, Alaska and other states, and snail fishes. Ingrid will take questions as we go, so feel free to just raise your hand or jump in as she talks. And thank you everyone for coming. And thank you, Ingrid, for sharing your work with us today.
So
thanks everyone for being here.
Yes, as Zoe mentioned, I'm happy to take questions throughout. I'm so happy you're here, and I'm excited to hear your questions and your comments and your criticism, anything. Unlike the cookies that are baked in the corner, my talk here is a little bit half-baked. It's a work in progress. And so part of my scheme was to give this presentation and work on it more after I get your feedback. So I'll be looking at the 10% rule for—
Are the cookies a reward for afterwards, or we meant to pass them around?
It's okay, whatever you like. I guess how do I go up? Oh, I got it. Okay, so the 10% rule of demographic independence was first put forward by Alan Hastings in a paper that he wrote in 1993. And it's a kind of informal rule that population geneticists and others use that says, if there's migration over 10%, then the populations are probably somewhat connected. And if there's less than 10%, then the populations are most likely demographically independent. And this rule has been cited a number of times. That I gave a couple examples here, and it's been cited in ways such as you see in this paper by McElhaney et al., where they say that substantial independence will occur when the proportion of a population that consists of migrants is less than about 10%. So
it's got m equals 0.2 feet. Like it was a good idea once and just has evolved. I've seen it so many places.
Yeah, you have good foresight, I think.
I'm leaving.
Why would I want to look at this rule? Well, in my role at the Alaska Fisheries Science Center, I'm often tasked with, giving advice on should these stocks be managed separately or perhaps together? And I put up here a bunch of motivating other papers that have said why it is important to understand your stock structure. Sustainable management, for example, should be based on knowledge of genetic population structure to avoid depletion of genetic resources. The task of assessing marine resources should begin with defining management units, and other statements saying that it is essential. But it is important. And here's a map of the region that I work in, and with some of the large marine ecosystems that we have, which don't necessarily always match the management units. And so we are systematically, one by one, looking at different species and trying to figure out whether the management units match the biological units. So if you start to think about populations and how they're connected, I drew this series of circles, and sometimes populations are just so close on top of each other that there might as well be one population. But the level of distinction can grow until you have clearly a distinct population. And usually I'm working in this middle area that's really fuzzy. And the main emphasis of this talk, as Andre guessed, is to look at how accurate this 10% rule of demographic independence is. And funnily enough, Paul Spencer, who does stock assessments at the Alaska Fisheries Science Center, met with Alan Hastings when he came to the University of Washington. And we asked him about his 10% rule. And he said, oh, I wrote that paper, but I didn't really mean for it to be used that way.
Equals 0.2 and 0.2b0, all on that same dash.
Exactly. And so I've always wanted to explore this, and I had some time recently. So
never read my paper on this topic, right?
You have too many papers to read them all. Send it to me later, and I would love to. See, that's why I'm here. Thank you. Okay, so say you're a scientist and you have this, you're tasked with understanding, are these populations separate or distinct? I put a list of some tools we have at our disposal. Every stock assistant scientist has access to data, for example, survey data that shows estimates of population size. And so you can always do a correlation analysis. Are these stock sizes looking like they're enough correlated over time? Also, you can grab a bunch of samples from each one, get a little money, and do a genetic analysis. There's tagging, and there's just observation. Often fishermen can give you a lot of insights. They'll say, I know those fish are from there, and those other fish go that way. There's something going on, and you can learn a lot from them. But for this talk, I'm focusing on two tools that will actually be incorporated into a simulation framework. How are populations correlated over time, and genetic analysis. And I want to give credit, first of all, to Robin Waples, who gave me the idea to look at correlation in population size over time. It's a really simple yet elegant way of kind of using data we have to see whether populations are independent. And the first thing I grappled with here is just defining migration. You might think, oh, 10% migration, yeah. But it's more complicated than that. You have a lot of life stages. It could be migration only at the larval phase, only in the mature adults, maybe even only in males. Is it one-way or two-way migration? Is it sporadic? Does it happen like a lot in 1 year and then none for 10 years? And then we also have to deal with seasonal migration in some cases. So I work a lot with Pacific cod, where it migrates in the summer, but then it comes back in the winter. And we know where it comes back to, but we don't really know where it goes in the summer. But needless to say, I'm not going to be tackling seasonal migration at all in this talk. If fish move, they're staying there in the models. So I also wanted to just kind of put out there that there's different ways to think about migration rate. So you have this number M that is kind of the standard way of thinking about migration rate. It's just the proportion of migrants. And if you were to think about M times your population divided by, okay, so your two populations, A and B, and you want to think about a migration rate between the two. So you could have the same migration between them, say 10%. A proportion of migrants in AP could be thought of as this migration rate times population A divided by what's left after migration in population B. And you could look at it similarly with your proportion of migrants from B to A. So I looked at just the strict migration rate and also this proportionality, because if you're thinking about it from a stock manager point of view, you might want to know what's the proportion of actual fish coming in relative to what's left after some left to migrate somewhere else. But there's a lot of other ways you could think about this. So in the models that I ran, I randomly selected migrants from spawning adults, and then I ran one-way migration and two-way migration. And in both cases, I just looked at not just the migration rate, but also the proportion of migrants in the receiving and the donor population. Migration was constant over time, and no seasonal migration.
Sorry, what are some of the fundamental risks? Because am I correct that you're assuming that if animal A moves population A to B, it becomes indistinguishable from B, which means if you move back to A, the probability can. So complete absorption in population.
Yes, and I did play with some parameterization differences between A and B, but it wasn't sophisticated enough that if a fish moves in a population with a certain natural mortality, this one, it changed its natural mortality. It just simply blended into that number there. Yeah,
Indistinguishable.
that was just kind of, yeah.
That's the same.
And then it's important also to distinguish that there are two kinds of populations. And I'm going to get into the idea that also defining the population is complicated. You could think of it as an ecological population. And this is a population that you think about it like demographically, which is really important in fisheries management. How many fish are there? And that's pretty much it. Are they moving and being connected or not? And then there's an evolutionary kind of a population definition where you look at reproductive cohesion. Are these actually interbreeding or not? Are they just exchanging so many migrants and not interbreeding that they look like one population? And it is a little bit challenging to think about these two when you're looking at genetic differences using genetics. You're usually in a realm where there's so little migration that the populations aren't correlated. And you'll see more results that support this later. And there is really no one answer to the question, what is a population? This is from Waples and Gagiotti, 2006. There's a lot of definitions for population. There's several for the ecological paradigm, several for evolutionary, and variations. And I'm not going to touch on all of them. I think we all kind of have an idea in our head what we think about as a population, but yeah, that also can be really complicated. So the simulations I set up, I used characteristics of Pacific cod and black-spotted rockfish. They're a nice set of contrasts because Pacific cod is shorter lived and really fast growing. Its distribution is pretty much everywhere in Alaska, so have ample opportunity to think about where they are and how they should be managed. And they also have this summer migration thing that really have to deal with, but we're not dealing with that here. We're just looking at this high natural mortality, fast growing fish. It matures about 5 years. It's been known to reach about 8 years, and its generation length, which is the average age from an offspring to when it matures and gives birth to its offspring, is about 8 years. Now, in contrast, we have black-spotted rockfish, and it reaches its maximum age we've seen at 147 years, and they mature much later in life, 19 years, and has consequently, much lower natural mortality. So this is a nice plot that shows the distribution of black-spotted rockfish. You can tell, so black-spotted rockfish were recently distinguished from rough-eye rockfish. They're a little bit hard to tell apart for sure, really hard to tell apart. But on this plot, you can tell where the black-spotted observations are because they're the black spots. And I'm going to be focusing on actually a region in the Aleutian Islands, the Western Aleutian Islands and the Central Aleutian Islands. Paul Spencer, you might have heard him talk about this, but he does the SAFS assessment for these Aleutian Islands, black-spotted rockfish. And there are definitely some issues which make this topic and this fish nicely relevant for this presentation. And the big problem is that in the Western Aleutian Islands, which you see registered by this blue line, the exploitation rate over time has been higher than the management quantities that we recommend. So this black dotted line is a tier 5 threshold for harvesting, and the red line is essentially like an ABC, acceptable biological catch. Whereas the Central Aleutian Islands estimates of mortality rate for black spotted is lower. It's much lower. So this is something that Paul has been grappling with almost since I've known you, Paul. So I'm doing some simulations. We'll see what we get. And because this has been a problem for quite a while, we and Paul have already done analyses to start to try and distinguish whether these Western Aleutian black-spotted rockfish are a distinct population from the Central Aleutian black-spotted rockfish. So there were 2 genetic studies, one where microsatellites were used, a second where actually we used low coverage whole genome sequencing, which is like the gold standard. You're looking at the whole genome, and we didn't find any differences between the Western and the Central. It would have been easy to deal with this problem if there was a genetic difference between these stocks. We can say, look, they're, they're adapted to their environment, maybe, or they're just distinct. So we have to draw a line here, but that wasn't the case. Yeah.
Just one basic question. Was there a reason why they were separately assessed to begin with?
Well, the whole Aleutian, actually, it's a Bering Sea and Aleutian Island stock of black-spotted rockfish. It's managed it's managed in that region. It's not managed separately.
But they're assessed, right? Like why?
They're not.
They're not.
No. And that the issue is that some people think they should be.
Well, I guess if we went back to the like why are they all in?
Yeah. This is an analysis, and you can speak up if you want, Paul. This is analysis that Paul did because he had a feeling that they were disproportionately harvested in the Western Aleutians, and wanted to point it out because it is an issue if there's enough differences between the stock and the rest.
It's a survey.
This is sort of
yeah, so this is showing the catch over the estimates of biomass from the assessment, which is a bigger space, and then it's partitioned out based on biomass.
Yeah, so this is kind of like an informal, they're not actually managed separately. And so they can go ahead and take as much as they want out of the whole quota for the whole Aleutians, right? Or the Central and Western. The Central and Western are combined.
Yeah, well, that's at the sub-AUC level, which are recent years of that problem. But the real issue is that it's about their stuff. They don't want the catches in some areas that are
So all these, there's so many blackfish are not migrating as adults. Would that be correct? So if they're being harvested there, there's thought to be low that because low for stock. So most of you produce something in it or something.
Yeah, we don't have any data. We don't have direct evidence of migration or not migration. But we do that we have compared them so many times.
Yeah, I will talk about that. So Paul did a management unit estimator analysis of the stocks where he restratified, essentially split into the Western, the Central, and Eastern Aleutian Islands, and found that the Western Aleutian Islands and the Central Aleutian Islands population sizes are not correlated. So based on this hope and management unit estimator, there should be a split there, right there where we actually have a separate area. So that's interesting. So we don't have genetic differentiation, no, and we don't have correlation, no. So what can we do with those 2 negative results? Maybe that'll help us move forward. So in the simulation, I kind of mentioned already, there's 2 populations, A and B. And there's either one-way migration from the donor to the recipient or 2-way migration. And I used an age-structured model where each fish was also assigned a genotype. So it's a age structure and individual-based model. But in the model, in the 2 populations, the fish grow and get fished, die of natural mortality, migrate, and some are selected to reproduce. And I have to say that I use a model that includes genetics. It's really unwieldy and slow to run. So I kind of separated out the population size aspect of the model. And in that, I was able to repeat for every migration rate level that I tested. I repeated it 100 times and counted how many of those times the two population sizes were correlated. And I quantified that. So, and I actually cut it off at 90%. So I said, if how many times out of 100 are these guys correlated more than 90% of the time? And to tell if they were correlated, I used the Pearson correlation coefficient, and I repeated those another 100 times to get some level of variance. It's dealt with each population. Yes. So each population has, we talked about SIGMAR.
Well, just how recruitment, one of the reasons for getting phase correlation is environmental, environmental variable spatial. And I
didn't add anything environmental, but I used variance on recruitment. It was actually about 0.4, but you do an independent draw in each population. So the populations were recruiting independently. So you'll see they have their own population trajectory. If it was the same, of course, they'd always be correlated.
Well, that's where you get phase correlation.
Yeah.
Where you get independent populations that are affected by common environmental.
They had their own recruitment variance each. They had their own dynamics.
That's as easy as
And they're otherwise
in some ways they were different that I'll explain in a second. Yes. And then yes.
Maybe you're answering here when I use replicate, how long you following
100 years. For this one, I just did 100 years. So and you can see what it looks like. Yes. So like I said, I separated out this part of the model that was only based on population size, and then the genetic model, it does have a burn-in of, there's a slide on that later that shows the number of years that I have the burn-in for the migration. And that one I run a little longer because I think it's good for that model. So, but for this just simply population size model, it was run just 100 years. It didn't need a burn-in or anything, but in each of these models, each population had its own independent reproduction, demographics. But so say I did one for cod and one for the rockfish. The populations had similar characteristics, similar mortality, some similar growth as you would expect. So this would be an example of a population where the population B starts out larger than population A, and they vary as they will. I measured that I let them be fished at ABC. So it was like, what should be considered ABC for cod and for
so it's equal F on the two populations.
Equal.
Okay.
Yeah, I didn't worry about how hard they were fished. They were fished, but at optimal levels. So here's my first slide of results. So I formulated this one for Pacific cod. And remember, I had one-way migration and two-way migration. And what I changed between the sets of populations was relative population sizes. And so here, this is kind of the key to the relative population sizes. And the first set is the donor, if it's one way, and the second is the recipient, if it's one way. So here's my one-way results, and then the two-way migration results. And you can see for several cases, equal population size, and when the donor population is smaller than the recipient, there was, in no cases was there any correlation. And if you imagine if the donor population size is small and you have one-way migration, well, it just tanked. So you didn't really have an opportunity for them to be correlated. There was much better results with the two-way migration. And it really got me thinking, like, one-way migration isn't even really realistic in nature. It's rare, I bet, to have just one population sending recruits to the next population. Maybe it is. They always talk about sourcing populations, but as I modeled it, I was like, I wonder how realistic this is.
You brought up all this very density dependent difference in the donor population and the same population sort of stay in equilibrium.
Yeah. Oh, and then this is key. So the scale here, where we have 0 to 15%, where I think I actually ran it to 20% migration rate. You can see that for equal population sizes and two-way migration, it's just about 10%, definitely in that range of 10%. But when you start to have different proportionality of the two populations, it shrinks. And here, this is also interesting that when you had the donor 10% smaller than the recipient, or the recipient 10% smaller than the donor, you still have, and it's two-way migration, the same, you have correlation at the same level. But it's pretty small, smaller than I would have expected it.
Right. The correlation coefficient goes to 0 or minus 1 to 1. So how are you deciding what is correlated? I mean, does it have to be positive 0.1 or positive 0.6 or 0.9?
I didn't look at like negative correlation at all, but I did a test, a p-value test to look at that correlation coefficient. And if it was under 0.05, I considered it significant.
So it was significantly positive.
Yeah.
Across 100 years.
Yeah.
Of the correlation has impact on the value to you can potential that you cannot find the value with correlation.
Is it possible that's built into the person's correlation coefficient?
That's one.
It's okay. I remember seeing that it's divided, like the variance and the variance are both in that. But no, I'll be.
It's certainly true that the higher the value of correlation, the better chance of it's significant. I mean, but you've got 100 years of data. I have to say, I don't think you've got 100 years of data. So you've got two things that you work in your favor to get high correlation. One is length of time series, and the fact there's no measurement error on the abundance. Whereas I'm suspecting the surveys for rockfish and evolutions have a little bit of measurement error.
Oh, so much.
So it's really optimistic in terms of detecting something.
Yeah, so maybe adding measurement error would be more realistic. Yeah. This is kind of just like one case example where you have a small donor population. You see it's replicated, and you can kind of see why they wouldn't be correlated. The donor decreases, stays small, and then this seemed important to me at first, but this is the same result, but now you're looking at migration rate relative to the recipient population. Because it seemed important at first, and then I kind of decided that I think we should just focus on migration rate. But if you were interested in, for example, how many fish are coming into a population, or what proportion of fish are coming into a population before it's correlated, it's higher. But in this case, these two populations are small relative to their donor. And in this case, this is, of course, measured relative to the recipient, and the recipient is quite big. So this one, I'm not quite sure about this. And here it is going the other way, where here you now have a really big, really small donor, and so it went the other way. Okay, so then now let's look at black-spotted rockfish. And you can see just overall, the scale is similar, but a little lower, a little less than 0.1 for the case of equal populations when you have two-way migration. And then you actually have no correlation in one-way migration. And I think that might be because the populations grow more, I'm not exactly sure. I think the populations grow more slowly. They're less able to kind of move in correlation. But there was no cases with one-way migration where you had correlation. Oh yeah, and this is just kind of comparing. We had a couple. And again, here's a one-way migration with black belly rockfish where they start out at the same population size, but the donor declines and the recipient increases.
Right. I would, this is Robert's point about burning, right? You know, that hasn't burned it. Like if you basically starting the population out in a non-migratory sense, then you're imposing migration and it's reaching quasi-equilibrium. But the reality is that happened that first 25 years happened in evolution.
I hear you. And actually, now that I remember, I started the correlation test after age 45 because that allowed 45 years of growth under this measure. And for cod, I started it after age 10 because that was the maximum model. So, and then here's a case where they start out as separate, very different population sizes, and the donor, which is larger, contributes, and they show some correlation. This would be a correlated result. Okay, so I've shown you what it looks like with one-way, two-way migration, different relative population sizes. But now I'm going to explore what sensitivity to natural mortality, maturity and age, and sigma R look like for both of these species. Here's, I'll just move the maturity age earlier, that's that. And then sigma R, it's variance in the recruitment. And if it's 0.2, if relatively small, you'll have more frequent changes, but they'll be kind of, the amplitude will be smaller. Whereas if you have large variance, you have these big peaks and drops in the recruitment. So I tested a high and a low of those. And here's what that looks like. So this might look complicated, but I'll go through it panel by facet by facet. So first is your base piece. So you've seen this before. That would be fixed, but you see that the pod, the two-way, and the one-way look familiar. Now let's look at different levels of natural mortality. So first I said, okay, in both populations, let's drop the natural mortality. It was 0.34, drop it to 0.2. Okay, then drop the donor or increase the donor population mortality to 0.4 and the donor and the recipient to 0.2 and vice versa. So you don't see much different there. Then the next three facets are changing early maturity to earlier in both populations, earlier in one, later in the other, later in the other. And finally, sigma R, lower in both cases. Oh gosh, this is a mistake. Okay. This is 0 because it is there. This is true. Actually, this might be true because I thought if it came up as infinity, it actually meant 0. So I went in and I thought I deleted all of those. This looks like it's an infinity that I missed, but this looks real. So that's interesting. So when you have differences in maturity.
I don't understand why maturity is affected. I would have put a priority that it's not going to affect but the maturity doesn't affect the dynamic. Who gets who needs to use it. Right? Yeah. I was wondering why you did it, but when I saw the result, I thought, oh, it's very interesting.
Okay, yeah. I'll look back at that one thing. And then sigma R. So, lower, higher sigma R. But overall, when I kind of just squint at this, I don't see big differences. And maybe I'll have to double check this maturity sensitivity. Mm-hmm. Yes.
When you changed the age of maturity, did you also change the burn-in time?
No. I can still get a bit like odd 10 years. Maybe that would be. Yeah. Off. Yes. Overton Holt.
So maturity might affect that. I think is
mm-hmm. Yeah.
Actually, the meaning of maturity, if you're moving mature animals, first in maturity changes actually N. The meaning of N as a population. That's the other thing. So be careful of that N hasn't been used.
Yeah, definitely. Okay.
And all you places is population size total.
So that one,
Is the population size you're comparing with the total population?
it is total. And I did it with mature too, and I wasn't sure what was the best.
I mean, obviously mature is a thing.
Yeah, and sometimes we are just concerned with the mature aspect of the population. That might be the most relevant.
When you use the real data, so that's
Yeah, I have that in my.
we're looking further on it wouldn't be mature. What it can see.
Which the other.
Well,
Okay, so this is the same kind of set of sensitivities for the rockfish, and again, there are no one-way results, but pretty consistent regardless of natural mortality, maturity, or sigma. Yes.
This goes back to that question for the definition. So statistically significant for the right, but that's over one applicant. 100 years, now 100 years. So when you say the lowest migration rate at which population size is supported, does that mean any of the 100 replicants that it can pass, or more than half?
So I'll walk you through. So I had a migration rate, and I would test the range. But say we're just going to start with like 0.05. So at 0.05, I'll run 100 times. I'll run 100 runs of these. I'll run 100 times these populations, and then for each one I'll measure. I'll count up how many is it correlated or not, and so it would be like say there was 80 times it was significantly correlated. That would get an 80, and then I would so for that set of 100 runs, I'll get it, and then I keep increasing the migration rate, and at some point I'd start to see 90s, and at the point where I saw 90s, I'd say, okay, that's the lowest migration rate at which we have correlation over 90%.
So that's lowest migration rate. Really,
so that one.
Lowest migration makes it consistent.
Yeah. Yeah. And then I would repeat that. And I repeated that 100 times again to get variance.
Oh, okay.
Which is kind of slow. Maybe it's my coding.
Okay,
yeah. That part gets complicated. And then I was like, try 90%, 95. And at first I didn't feel like I was getting much correlation. So I was like, I'll do 90. So I can go back and change all this. Okay, so I said that there's also an aspect of the model that will do, we'll take individuals from the population and test them to see if populations are genetically distinct or not. So, you know, I had my two populations, A and B, and I would randomly select some males and females based on the number that population dynamics model said were spawning. And I would take those adults, and each one was assigned a genotype. And I had some computational limitations. That is definitely a little plea for more computational power. I'm looking at Melissa. And I was able to use 50 SNPs, which is, it's kind of on the smaller side, but that's what I had the power for. And so each individual had a genotype of 50 SNPs. And then I would randomly select by Hardy-Weinberg Mendelian inheritance genotypes for the often. And that's how it worked. So I had an evolution period that was 100 generations where a population would evolve with limited dispersal. And then a simulation period where each of those fish in the populations would perform as they, you know, they grow, reproduce, die for 300 years. This part also just, I think that I need to do more of them, and I need to do just bigger populations. I was limited in that too, but I got some results. And so this is what the result of those models look like. So I'm going to give you just a couple outputs of that, and I'm just going to summarize because it gets a little much. So I'll be talking about a measure called FST, which is a measure of genetic differentiation that goes from 0 to 1. And if it's high, the populations are really different. And if it's low, they're really small. And what I used to tell if they were significantly different was I did my draw of 100 from one population. I did a draw of 100 from the other population. I did the FST test. And then I permuted that 100 times to get a significance level. So for cod that have equal population sizes, and here's the population size total. It's only like less than 30,000 on average. And in this case, I did one-way migration at of M of 0.001, and these populations were equal. That's what I called proportion 50%. You can see this is the burn-in period where the populations were simply exchanging. So it's like one migrant per generation, and then here is where. Okay, that's 200 years. 200 years of fishing and population dynamics occurred. You can see what the pairwise FST between each of these sets of two populations, and I did 10 replicates here, what FST looked like. So it's between 0.3 and 0. And here's the significance here for 10 separate runs I did. So for this scenario with M is 0.001, all but one of my separate runs were significantly different based on this FST test. Any questions? I know that's really a lot.
So the two populations started off with an FST before?
I just generated sets randomly, assigned them randomly, and then when we run for 100 years, some migration between them, it starts to block. They become more similar.
So I think the different colors are different
runs, like different, like I simulated this exact same thing 10 times. So here's your first repetition, replicate it up to 10 times.
What's the weird? There's something weird
going on. I think there's some weird blip where I all of a sudden taking these phenotypes and assigning them. I kind of changed the format of how the types are spread out among individuals there. And here's kind of looking at it a different way where, okay, let's look at the scale of FST here. So FST is like up to like 0.5, and you have this low level of migration, but now we increase migration to 0.05, and you can see that with higher migration, FST drops. It's much, much lower. And indeed, significance is no longer, it's only in 2 out of 10 cases. So now we can kind of start to compare where we were with our demographic model and then a genetic model, say, okay, would these populations, if you had 50 SNPs and you sampled 100 fish, would these appear to be different or not? And so the threshold here for these 2 equal population sizes with one-way migration ended up being at about or a little less than 0.05. So now I'm going to put the demographic results together with the genetic results. So this was for our migration rate for cod, at which we had the lowest migration rate at which population sizes are correlated. So say here for your equal populations, the migration rate for equal populations is correlated at about a little over 10%. So with genetics, you pick up differences between 0.03 to 0.05. And when the populations are different in size, I noticed that you would get genetic differentiation significantly at a lower level. And there's still some work to be done. Like I put less than this because I wasn't quite honing in on exactly where it was at. Okay, so let's go to black spotted rockfish now, where I did my best to mimic everything about the population sizes that I could to try to bracket where we think the amount of migration we think is going on between these 2 populations. So the Central Aleutian Islands stock is bigger than the Western Aleutian Islands stock. And I took the mean and averaged it over this time series. And the Central Aleutian Islands stock is on average 5 times bigger than the Western Aleutian stock. So I gave it about 5, I modeled it as 5,000 tons, and the Western Virgin Islands stock as 1,000 tons. And when I did that, I did remember in the sensitivity test, the minimum level of migration where you have correlation changes with relative population size changes. But in this case, all I did was change the absolute population size, and it was 0.04 across the board. And genetically, it also appeared, I actually, honestly, I couldn't go this high, so I should probably keep the stars up. I had done other studies where it looked like, I just should keep the stars up. So ignore those stars on the left. But with smaller population sizes, the level at which you start to see genetic differences is really small, somewhere in the order of 0.001. And the order that you see demographic differences are about 0.04. So that was my best guess at the migration rate between the Western Aleutians and the Central Aleutians black-bellied rockfish, which to me, that's pretty small. So also, I would conclude that the migration isn't all in one direction. And I don't know, and I can't say anything about whether the migration is constant or stochastic, but likely it's stochastic. And it was really only relative population size that made the most difference in the results. So for our next step, I think it would be interesting to take these kind of bracket this migration rate and look to see if it might be sufficient to counteract the effects of fishing. And it kind of leads me to this definition I've seen for a definition for a distinct stock. That's something I want to come back to, which is that it could be a group of individuals that are isolated enough that immigration or migration in does not substantially affect the population dynamics over a 10-year time frame. We can kind of think about the results for the black spotted part of the analysis in that way. And also it kind of back to the ecological paradigm, which is the evolutionary paradigm. It's important to remember that the demographic decoupling stops when immigration stops, but the genetic differences will reflect historical gene flow. And that's my last slide. So I don't think the 10% rule is simple, but I definitely think that it's more complicated than just that, and maybe applying the simulation to questions you might have could be useful to get at migration. And also, I want to say that in science, it's good to question paradigms and question things that you might take for granted. I got that idea from my sister, and also the cookie recipe. And I also wanted to thank Robin Maples and Professor for their help on this. And I'm ready for questions.
Get some more migration.
Is it Albie?
Yeah.
Albie.
Is there any information at all about large or dispersal like between these two areas, like currents and stuff, especially for things like rockfish that have pretty extensive as far as I know in the Atlantic state before they even settle and then actually move.
So I think that newly extruded rockfish larvae are totally unable to decide where they go. But I was able to work on a study where we had this kind of a plankton pump that went into a coral field, and we found larvae that kind of take refuge in coral. And also they pulled up some coral and they found all these tiny larval rockfish attached to the coral. So I don't know, but that might be a mechanism that larval rockfish aren't moving everywhere willy-nilly, but there's got to be some that move just by the currents. I know that adult rockfish typically don't move far, but I don't know.
Who's not? Couple of things to think about. The first thing is, what does the 10% migration rate mean?
Yes.
And so when people have done this to me, firstly, I always think, well, surely this must depend on tax. And why do I say that? So let's take a few populations. you're using 10% per year. That's for the moment, say it's one-way migration. So start with a unit of 10,000 animals. First year, you lose 10% to the other. You got 90% there, right? 1 minus 0.1 to the power n is the proportion of animals that don't move, right? For a spotted rockfish, 107 euros, 10% migration rate means that every animal will move, right? So we need to think here, that doesn't sound like small migration. So you got to think about how many years that animal is around. So if it's a sardine, a 10% migration rate on an annual basis is very different than 10% for a long-lived rock. But first thing I think. The second thing I tend to think about is I find this argument sort of the wrong way around. You're saying, how much migration do I need to detect? Right? I don't care about that. What I care about is all time, which is, does it matter that F is higher in one region than the other? Right? So what and I never took this as far as I would like, I just said, like the 10% rule. This is the answer, believe me. I thought much simpler than hardware. But essentially, the M we care about is the M such that if you fish higher in one area than in another area, you will end up with local depletion. Right, so it's basically, and when I did this, I think, Robin, I think I said this in the waiting meeting. It's something like, sorry?
Probably, but then you have courage, you have to have a definition.
You said, well, yeah, I think it was depletion below the tolerance or whatever, the halving of it. But you can, essentially, it's a function. It turns out that argument, and I agree with Robin that exactly how far you go down depending on a definitional issue. It's related essentially to FMS one. So if you've got two populations with high, if you're fishing, let's just take it simple. There's two areas, you're fishing one and not fishing the other. How much fishing can you have in the second area relative to FMS5 such that the immigration doesn't balance it out? Right, you've got immigration and then you've got F. And so essentially what I was arguing is once the migration rate is less than FMS1, that's when you're starting to be in trouble. Because the migration can't keep up with the fishing.
You mean once migration is greater than FMS1?
Otherwise, because remember that you're fishing too hard in one area. Right. So if you're fishing too hard in that area,
less than
you want productivity coming in such that you don't deplete the source population by sucking them out of the salt sink population. So thinking about, I guess what I'm saying is from a management point of view, I think about it as, can you sustain over harvesting in an area because of migration, rather than can you detect migration if it happens?
Oh yeah, and I have this all set up that wouldn't be hard to put that together.
So basically, does Paul need?
I hope you don't, Paul.
Okay, but I'm going to get back to you were history. Certainly, I think those arguments really likely but I think you took it as got really basically there's no real warning practice coming out of the already from that identified population. But because this is what we actually want to know. And so you just do this exercise every practical situation you come up with. And if it doesn't make a difference, then there's no problem in the area. There's not any problem on the calculation of food. And if it does happen, then you need to go to management to take account of whatever your conservation objective is.
That's pretty good. Now you're reminding me of what I was talking about. So what I think I did was I said, you've got two populations. The exploitation rate is higher in one than the other. Sorry, two areas. You don't know the population. Exploitation rate is higher in one than the other, right? So what you then do is you say, given that, what is the critical value of M such that you can sustain it? Then the question is, can you detect it from, for example, the data that you're talking about here? So it turns it around from what M can I detect to can I detect the M I care about? And if you can't detect the M that you care about, then you go down Robin's road, which is if you use precautionary management, then maybe you need to spread the effort simply because if there's a problem, you wouldn't have the power to detect. So sort of turning it on its head and starting with the management problem and then asking the monitoring problem rather than starting with the monitoring problem.
Yeah, for this fact, the F1 slide would be an order of the people, but yeah, I have to agree. So I think you're basically still right.
That
yeah,
migration is a little higher than FMSY, so it doesn't matter.
No, it's to be lower. You know, so I thought the range of migration.
Oh,
okay.
Yeah. Well, then we're splitting hairs. Is it lower?
Right.
Yeah.
And you're fishing right there. There's my analogy. It's simple. So I would encourage you to take that into that management.
No, I think that it sounds like a really good idea, but I actually think we're going to be still in the I don't know range, right? Because we're kind of in the middle of it.
Yeah, I mean,
depending on what FMSY is.
You know, that, I mean, that's where colleges are a little bit higher than the children. I think we made that more recent study. Yeah, because like Kevin's probably an order of 1.4, 1.45. That's what I'm around 1.4. So would be, you know, if the range of vibration are right.
Okay.
You're the 1.4.
Yeah.
You know, that's a lot of we have to go high.
Okay. Okay, so this might be interesting. Good ideas.
I always wanted to work on this and I never had to get to finish it. So
well, maybe we can work together.
Well, we should probably wrap up because of the class coming in. But feel free, I'm sure Ingrid is open to further discussion and email communication. So feel free to reach out to her. Yeah, thanks everyone for coming. Come back next week to see Leachy.
Yes.